Welcome to Birch Bioinformatics. My name is Brian Frostensky. What I want to talk about today is all the things about computers that you need to know if you're a biologist just starting out in bioinformatics, but may not have seen before. My experience in teaching bioinformatics has shown me that if you've only done the standard sorts of things with computers like web browsing, writing documents, spreadsheets, and so forth, there are going to be a lot of gaps in knowledge. These can be little things like what text files are, or big things such as how to run a session on a high-performance compute server. So we're going to start, first of all, by describing Linux, which is an offshoot of the Unix operating system, and this is a system specifically designed for scientific computing. We're going to make the point that you can do just about anything you want on a Linux server, and also why there would be big advantages to that. If you're going to work on a server, of course, you need to know how to move files back and forth between one computer and another, or your desktop computer and then the server. So we'll go over a bit about that, and we'll also make a big point about organization of your files. You're going to have hundreds or thousands of files, and you simply can't function in bioinformatics if you don't have an organization strategy. The other thing that a lot of people don't really learn how to do is take advantage of their screen real estate. On desktop computers, we have really large monitors, and very often you'll have one program taking up the whole screen. We'll see how you can get the most by using multiple windows and doing different tasks in different windows. Finally, I want to make the point that programs are tools and the computer is the workbench. If you think about it, you've got maybe hundreds of different bioinformatics programs available. You might want to use these in different combinations to do a complex task. This is the kind of thing that we're going to talk about, which is often known as data pipelining. So I'd like to start by talking about why Linux is often the system of choice for scientific work. Of course, it's a free operating system, which is nice and easy to use. Just like any desktop environment, you have basically the same types of programs such as web browsers and office productivity suites and so forth. But there are a lot of things that make it particularly useful for science, and one is that it was designed from scratch as a multi-user system. The idea is to make as full a use of machine resources as possible in as efficient way as possible. This makes it possible then to have a fairly complex bioinformatics system running on a Linux server that can be used by many people, rather than trying to get all of those things working on dozens of individual PCs in a department. So Linux is very efficient. It makes use of the machine resources to the maximum extent possible. It's intrinsically secure. It's designed to be a secure system because it's designed to have multiple users. Networking was always a part of the design. And then finally, there's the Linux command line, which sounds like an antique kind of a way of doing computing, but actually turns out to be far easier for some tasks than the typical point-and-click software that you may work with. If your lab or department or institution is trying to put together a bioinformatics system, you really want to put together some kind of a Linux server. Maybe one final thing to add before we get off of the topic of Linux is that because Linux is so efficient, it can be used to rescue the most obsolete machine. And just as an example, this entire presentation, as you see here, is being produced on a 12-year-old Mac, and this Mac couldn't be upgraded to any of the recent Mac OS versions, and so the only thing that was left to do was to wipe the disk and install Linux, and then I'm able to do just about anything that I could use a normal desktop computer for, and things work actually quite well on these type of Linux systems. So my philosophy is, why buy a new computer when you can get the same performance by switching to Linux? Now, most of you have only used desktop computers or laptops that are designed for a single user at a time, for just use by that one person. And typically computers like that are designed around the everyday things, browsing the web, looking at videos, writing documents, and that kind of thing. But for bioinformatics, we need to get beyond the limitations of the PC. And in fact, it's often much easier to set up a single server that has all of your bioinformatics software on it and configure everything properly so that it works for everybody who uses the system. Trying to reproduce that on every single machine in a department or across an institute is a tremendous amount of work, and not all is usually ends up being an endless source of problems because each computer is really a special case. So by offloading things onto the server, as we'll see in a moment, we actually drastically simplify the process of getting bioinformatics software out to people. Um, in bioinformatics, transcriptomics or genomics tasks often have data sets of maybe 25 gigabytes, 50 gigabytes or more, and this is a substantial amount of data to handle. In some cases, say a genome assembly, you might need more than 100 gigabytes of RAM just to do the assembly, and that's way beyond what a typical PC has. Typically, servers also have a large number of central processing units known as CPUs that can break down a task, again, say like a genome assembly, into smaller parts that can all be run simultaneously. And then finally, the other thing that is different about a server-based system is it's always on a hardwired, high-speed internet connection, whereas your desktop computer or laptop is typically on Wi-Fi. Some don't even have a cable port anymore, and so despite what the advertisers claim, Wi-Fi can be flaky and it's not always reliable, especially when you're trying to transfer multi-gigabyte files from one place to another. So for this reason, in bioinformatics, we typically want to go with more of a server-based system that you can access from almost anywhere. The motto of Sun Microsystems used to be, "The network is the computer." And I think that's a very apt description of what we're trying to accomplish here, to offload to the high-performance computing systems most of the work that your PC can't do, but leave the display part on your computer. So we can think of the diagram here as showing how the tasks done on a computer can be broken into different layers, which are done in different places. So for example, when you run a session on a Linux server, that's on one of your servers, and at the University of Manitoba, the way we have things configured is there are numerous redundant Linux servers. You can log into any of them and do basically all the same things, because there's another layer called the file server or file services layer, which is where all of the files reside. And so, in effect, the file server acts like a big disk drive that's mounted onto the each of the servers, the Linux servers, as if it was just another disk drive, except that happens across the network. So, for example, here we have the home directories of users named Bob and Carol and Ted and Alice, and we also have things like the GenBank DNA database and programs for bioinformatics, all residing on the file server. So it doesn't matter which of these Linux servers or login hosts you are working on, they're all reading files off of the same file server. So all of that is happening off on the server or in the cloud, if you want to call it that. And the one thing that's happening on your computer at home or in your office is the display part. And so here we have different users, Alice, Bob, Carol, Ted, all logged in to their accounts at the same time, all doing different things, and they'll be spread out onto different servers to spread out the load. So the net result then is that you are being able to see what's happening on the server and have that display on your screen. And the way that works is that there's an image of the screen that's called the frame buffer, and that is always updated anytime you move a window from one place to another, or scroll up and down through a window, or just move the cursor. And so the program then that's running on your desktop computer is called a thin client. ThinLinc is one example of a thin client program. And what that does is it keeps track of changes happening to the screen on in memory on the server. So anytime a window moves in the frame buffer on the server, that same window will be echoed, so to speak, on the copy of the screen that's on your desktop. So this means really that any user can do any task from any computer. As long as you have a thin client software, that one program on your desktop machine, you have the ability to do everything that you could normally do on the server. So this is a very powerful capability, and it means that you can work from anywhere and do just about anything. So next I'd like to demonstrate a program called ThinLinc from Sendio.com that runs a session on the remote server but displays the session in a window on your desktop. And so this is a way of leveraging even the oldest PC, no matter how low powered the machine happens to be, you can have the full capabilities of the server in essence on your desktop. So to show you ThinLinc in action, I'm first of all going to show you my desktop, and so I have the presentation you're looking at right now in the LibreOffice Impress presentation program, and then in this window is a session that's running on the remote server at the University of Manitoba. So the program that's doing this is called ThinLinc, and so anything inside the ThinLinc window is happening on the remote server. Now I like to have a lot of space on the screen, so I'm going to change this to full screen mode, and now you can see I've got the full set of applications running on the desktop. So in one window I've got the manuscript that I'm writing, and then here's the reference document, or the references for that manuscript. And in this particular paper I'm wanting to give examples of the biologist software running, and in this case we're going to do a search at NCBI for any proteins whose name is squalene synthase coming from algae. So I just have to click on run to do that search, and a number of proteins come up, and if I wanted to retrieve these proteins I could just select them and then run the protein fetch to retrieve them to a new biologist application. This time it will have all of the proteins in it that were found by the keyword search. So this is just an example of running a remote session. Once again, if I go back to the smaller window, everything that's happening on the server is displaying inside a window. So this means, again, any user can do any task from anywhere. All you need to install on your PC is the ThinLinc client, and you can run a session on the remote server. So ThinLinc is one of many different thin client programs that are available. There are others at the University of Manitoba. We have a campus-wide license for the ThinLinc server, and so that's what we run on our system. I will point out that the ThinLinc server is actually free for 5 simultaneous users, and surprisingly cheap if you go above 5 users. So even for, let's say, a departmental machine or even a machine in a laboratory, the annual fee is actually quite affordable. And so this is a really good way to leverage the power of whatever server you happen to have. So this means that you can access your server even when you're away from campus or away from home. And so I have here a kind of a whimsical picture of being on vacation in Florida, and having ThinLinc on your laptop is almost like having a really, really long cable going all the way back to your server. But this will work in hotel rooms or conference centers or libraries or just about anywhere where you have some kind of a Wi-Fi connection. And so this is a very good capability to have, and I've found it to be tremendously useful even over long distances. So one question you may be asking yourself is, if I'm working on a remote system, how do I move files back and forth? And there are a number of ways to do this. What I've found to be one of the easiest is a program called FileZilla. This is an open source program that is freely available for download, and I'll just show you an example here. So on the left side we've got the folders on the desktop computer that I'm using right now, and I want to upload some of those files to the remote system. And so that the remote system is in the right hand side. So I just have to select the files I want to upload, choose upload, and you'll see the progress as these files are uploaded. Now these are video files. These are actually the video files that are part of my presentation right now, and in real time we're seeing those being uploaded. So this is generally a pretty quick way to move files back and forth from one system to another. And there we are, we're finished now, and so you'll see that all of those files are now on the remote server. So it's that easy to move files back and forth between systems. So I want to move now to a topic that again is something I think many people don't really learn when they're doing regular desktop computing, and that is the concept of a home directory. And so all systems have it. Windows has a sort of a default directory that things will get stored in, and on Macintosh you'd have /users/ and then whatever your username is, and in principle that this is a place where all of your files are stored, and that's where you work. And on the personal computer platforms, this is not always strictly enforced, but in Linux and Unix, this is very highly enforced, so that the only place you can save files and store files and change things is in your home directory. Now, by the way, the word directory is synonymous with folder, so if I say directory, I really mean folder, and I may use those two interchangeably. They're synonymous. And so on any Unix or Linux system, you'll have a home directory for each user. So I'm showing here an example where the home directory for a user named Homer is in /home/Homer, and another user named Bart has a home directory called Bart inside /home. So his home directory is /home/Bart. Now, when you start a session or desktop session on a Linux machine, you're automatically working in your home directory. But then you can create subdirectories or folders. So in this case, Homer has a directory called beer, and another one called donuts, and another one called nuclear waste. That's how he organizes things. And then Bart has a directory called skateboard, another one called TV and comic books. So each of those folders contains things that are related to, let's say, skateboarding or TV or comic books in the case of Bart. Or at least that's what he wants Homer to think. He doesn't tell Homer what's really in them. So by having all of your files and subdirectories in a single home directory, that first of all is a great organizational thing. It separates your files from everybody else's, but it also is a security feature, because the only things that Bart can access are within his home directory. The only things Homer can access is within his home directory. So now I'm switching on to the XFCE desktop that we have running on our campus Linux system, and if I open up the file manager, you can see a list of directories. And so just for example, I'll take a look at my research directory. So if I go to my research directory, I have subfolders. There's one for grants, so when I write a grant, I have a separate folder for each grant. There's my current NSERC grant, and all of the files related to that grant. And so if I wanted to take a look, for example, at the references, I open up the reference document, and there's a PDF of the references used in the proposal. And so any file related to the proposal is in this folder. And then maybe I could go up one or two and take a look at the projects. So any current project that's going on, or even some of the older projects would be there. Things like my CV, different CV folders. So just about anything that you're going to do. If I review a paper, For any paper, I've got a separate folder. So for whatever I'm doing, there's a folder. And this is so important because in bioinformatics, we end up having each project might be a folder, and then you would have sequencing reads, and maybe the raw reads would go into one folder, and the processed reads might go into the next folder, and maybe you create some BAM files for an RNA sequencing project that goes into a separate folder. So everything we can do should be organized into folders. And I know there's a temptation to put things on the desktop. That ends up being actually a counterproductive thing, because pretty soon you've got all these shortcuts and you don't even know where the actual original files are. So I'm going to make this a short point and just say that trust me, you need to organize everything into folders. And maybe one other point, you can see that I'm using what Windows would call the detailed view. I know that you can have an icon view. This is a pointless thing. All it says is there's a folder. Why not actually see some detail and see things like dates, for example, when a file was created? If you had a zero length file, that might be an indication that something is wrong. So the more information you have, the better. And this is why I would always use the detailed view rather than, let's say, the icon view. So the next topic I want to cover is best summed up in this question. In the 21st century, why do we still need a command line? And so in the olden days, before most of you were born, that's the way computers worked. As you typed a command, the computer ran a program, and it gave you the results by typing text out to the screen. And this sounds like a dinosaur kind of way of computing, but it is interesting to note that in the Windows has always had sort of the DOS command line that you'd be surprised how much use that actually gets by developers. And on the Mac, in the when Macintosh shifted from the Mac OS 9 to what they call OS 10 now, or Darwin, they actually threw away almost all of the Macintosh code and put in a version of Unix so that there would be a command line. And so these are examples of why many people have thought that the command line is useful, and in fact it is, because we need to be able to do things in a more automated fashion than you can do by pointing and clicking. So one of the reasons a lot of people like Unix and Linux type systems is they have the Unix command line. And just to illustrate some of the power of the command line, I have an example here of some output from a blast search. And so the output is giving us the name of the database that the search was done on. So this is GB, stands for GenBank. And then the next field, the field separators are these little pipe characters. The next field is the ID number of a hit. And then we have a short title of that hit to give us an idea of whether or not we want that sequence, and then a couple of other fields, I think this is the raw score, and then the e-value, which is the probability of seeing that hit in the database just by random chance. And so if you wanted to retrieve the sequence hits that were found in the blast search, it would involve somehow dissecting out these ID numbers so that you could then do a batch retrieval of all of the hits. And that's very hard to do, let's say, in a text editor, but it's very easy to do at the command line. So I'm not going to go into all of the details of what this command line means, but suffice it to say that on a single line, just by typing a single line, you can read in this blast file, cut out the second fields when you're separating fields by these two pipe characters, and then send the output into a new file that has just the ID numbers, or the accession numbers actually is what these are called, that you could then use to retrieve the sequences. So the command line is very powerful when you need to automate things or when you need to do things repetitively. The more you have to point and click, the more mistakes you're going to make. And so the command line actually protects you from making those kinds of mistakes. Now later on I'm going to say more about the command line and actually do a very short demonstration of how the command line works. But before I do that, I want to get across a couple of other concepts that people may not really know a lot about, and that is what do programs do is the first one. So I like to compare a computer with a cell. And so since I'm assuming most of the people watching this video are biologists, I'll talk first about a cell. So in a cell, you know that there are biochemical pathways, and if you have ever seen one of those charts that hang on a wall that has very fine print and all these pathways, the TCA cycle and the Calvin cycle and all these other things, glycolysis and gluconeogenesis, and you go on and on about the different pathways, the anabolic pathways, the catabolic pathways. It would be surprising to learn that actually all of that complexity can be summed up in a single rule. An enzyme takes a substrate and produces a product. And from that one simple rule, you can build the most complex pathway charts you can imagine. So you start with one molecule, the first enzyme takes that molecule and does something to it. The next enzyme takes the product of that reaction and uses it as a substrate to produce yet another molecule, and then the third enzyme might take that product and do something with it and produce something else. You can even have branch pathways, and so all of that complexity really is summed up by this simple rule. Well, the same thing happens in computers. Here's the rule. A program takes input and generates output. And there's no reason that that output couldn't be used as input by some other program. So from that then we get the concept of data pipelines. So let's just say as an example, you start out with a genomic sequence. That's your first type of data. There's a program that will extract protein coding sequences from the genomic sequence and give you now a file of protein coding sequences. Maybe there's another program that knows how to translate coding sequences into proteins. So now you've got a file of proteins, and then the third program might take those programs and do some protein task on them, maybe predicting secondary structures or calculating amino acid frequencies. And similarly, we can have branches in the pipeline that maybe the coding sequences are have something else done to them, and you produce some new output there. And so this kind of a chain of programs is what's referred to as a data pipeline, and for anything other than the most trivial tasks in bioinformatics, you'll be utilizing numerous programs in some kind of a pipeline. Another concept that comes along with this idea of using programs is the idea of text files. And so up to now, let's say if you were working with a spreadsheet, it was probably an Excel spreadsheet, so you had an Excel file. If you're working with a document, it was probably a Word document, or if you're using LibreOffice, it's a LibreOffice document. But one way or another, the only program that can read that file is the program that produced it. So Excel can read Excel files, Word can read Word files. If you had a graphic bitmap graphic, let's say a JPEG file or a PNG file, you might have a viewer that could read that, or maybe you bring that into something like Photoshop. For program input in most scientific applications, that's typically done with what are referred to as text files. So a text file, think of it as a document that doesn't have any of the special formatting. There's no fonts or boldface or underscores or pagination or footnotes or headers or whatever special features you have in documents. It's just plain text. And so the beauty of that is that you can open a plain text file with a text editor. So a text editor is like a word processor, except that it reads text files. And regardless of what the text file is, you can read it with a text editor. There's an example of a text editor that's read in an alternative genetic code. And so this happens to be the genetic code for flatworm mitochondria, which is slightly different from the universal genetic code. For example, here, TGA normally is a stop codon. In the flatworm mitochondrion, that codes for a tryptophan. TAA codes for a tyrosine. So the only stop codon in this genetic code is TAG. So you might have a program that translates DNA into proteins, and so you would need to be using the correct genetic code for the organism that you're working with, and so your program might need to read an alternative genetic code from a file. So most input for scientific computing is done with text files, and then the output will usually also go to some kind of a text file. But that brings another point, which is that when you have a text file, you can look at it. If you don't know what's in it, go ahead, open it up in the text editor, have a look at that file, and see what's in it, and that will give you a better idea, perhaps, of what you're supposed to do with that file. Another in my list of things that you don't always quite know, or maybe a sort of know, has to do with the distinction between files that are on disk and files that are in memory. So you know that the memory of a computer is where things happen. That's called RAM, or random access memory. And let's just say, as an example, I want to work with a document. And that document is one of many files that are sitting on the disk drive. And let's say I want to work with document 2. So if I open that document, the first thing that happens is a copy of document 2 gets written into random access memory. And it's in RAM where I actually work on it. So the whole time that I'm looking at the document and scrolling up and down and making changes and doing all those things, that's a copy of the document sitting in RAM. But the consequence is that the original document is still in its original form on the disk. And it's only when I press save that the copy in memory gets written onto disk, and now it overwrites the original file. So once save occurs, whether it's an autosave or whether I press the save button, one way or another, that's the only time that the copy of the file gets updated on the disk. So one of the consequences of this is that in some cases you may have say if your program crashes, you might lose the copy that's in memory and you'll still have the original file that's on disk. Hopefully that won't happen, but this is also I guess implying that you need to save your work onto disk, otherwise it goes away. So this is something I think most people know about, but they may not know exactly what's happening under the hood. So now I'd like to actually demonstrate the command line, and since we're doing something completely new, I'm just going to switch to Workspace 3 where I opened up a terminal window. So maybe just to demonstrate that, I'll go back to the Applications menu and show that you can access the terminal window from the Applications menu, and the other place you can do it is I've created a launcher on the top bar on my screen to launch a terminal window. And I'm going to make the font bigger so that everybody can see easily what I'm doing. And so the command line is a way to tell the computer I want to do a particular task. And so the simplest type of command might be to list the files that we have in the current directory. So if I type the ls command, that says give me a list of the files in the current directory. So there's a lot of files in this directory. In fact, most of these are actually directories rather than files. The ones in blue are directories, the ones in white are files. And So if I wanted to see more detail, I could type ls-l. And that's the ls command using the L function, the long, asking for a long listing of the files and directories inside my home directory. And so now I get more details on the files that are present. Now, what I'm hoping you're getting right away is that I'm able to type in this window a command and then get a response. And every time I type a command, I get a new prompt. So this thing in brackets at the bottom is called the command prompt, and I've got the command prompt set up so that it tells me the name of the machine I'm on. I'm on a machine called Mars, and then the current directory where I'm working, which is my home directory, and that's in /home. There's a subdirectory of /home called plants, and then inside that subdirectory is my home directory, and that's called FRIST, the first 5 letters of my name. It refers to my home directory. And so every time I press enter, the enter key on the keyboard, I get the prompt back. And so the prompt is telling me that the shell is waiting for me to type a command, and then it will try to execute the command. So the shell is a program also. In this case, it's a program called bash that reads the command that I type and tries to figure out what I want to do, and then executes the command. So there's a command called ls. So if it sees that with nothing else, it will run the ls command, and by default, it will show me the list of files or directories in the current working directory. And again, as I said, ls space -l will give me the long listing. So now we're seeing an example of the fact that when you type commands in Linux, you need to separate the different parts of the commands using blank spaces. So ls-l gives me the list. If I typed ls-l without a space, it's going to tell me command not found. There's no command by the name of ls-l, but there is an ls command, and the -l option is going to give me a long listing. So ls-l with a space between the name of the command and the long option will give me the long list. So we're going to just try a few more commands. I'm not going to attempt to teach you all of Linux right now, but what I want to do is just give you a flavor for how commands work. So every command does a different thing. There's a command called mkdir that stands for make directory. I'm going to create a new directory, so I'll type mkdir space, and then we'll call it demo. It's a new directory called demo. So now if I type ls, you'll see that there's a new directory called demo, and if I type ls-l, we can also see the demo directory is created right now, and so there's the timestamp for it, and it also tells us something about the permissions for the files. We'll talk a little bit about file permissions, I guess, later on, but right now we know that we've created a directory called demo. So if I want to go inside that directory, I type cd. CD stands for change directory. Go to a new directory, and what directory do I want to go to? I want to go to the new directory called demo. So you'll note when I type cd space demo, first of all, of course, I have a space between the name of the command and this argument that's sent to the command. I have to tell the cd command which directory to go to. You'll notice that the prompt has changed as well. So the prompt on our system, at least, is set so that it tells you the name of whatever directory you're currently in. So we've by typing cd demo, we've moved into the demo directory. If I type ls-l now, you'll see that it says there's a total of 0 things in this directory. Demo is an empty directory because I just created it. So okay, maybe we want to create some files to put into the directory. We can do that using the cat command, or the echo command rather. So the echo command is a command that prints back whatever you type. So if I say echo echo something or other, it prints back the words something or other. So anything after the word echo is printed out. So remember, echo is the name of the command, and then everything after that gets printed out. So that gets us part of the way. I said I wanted to create a new file, and one of the things that Linux lets us do is redirect the output of any command to a new file. So let's try that. Echo something or other, but now I'm going to use this right arrow character to send the output to a file, and I'll call it something like file1.txt. TXT, I'm putting the extension on to indicate that it's a text file. And so maybe before we go too much further, I always like to show how in many cases you can do the same thing in the file manager as you can, or with the graphic interface as you can with the command line. So I've opened up a copy of the file manager, and you'll see here that the demo directory is present. So if I double click on the demo directory, there's my directory with file1 in it. And so I could do the same thing here. If I type ls-l, it shows me now that file1.txt is in the current directory. So if I wanted to, I could easily go back and forth just by moving the mouse from the terminal window to the file manager, depending on whether the task I want to do is easier to do with the file manager, I'll do it there. If it's easier to do with the command line, I'll do it in the terminal window. This is a great convenience because now we have the best of both worlds. We have the point and click capabilities of the graphical desktop, but we also have the ability to go to the command line at will. So let me create a few more files. Let's say so now I'm going to print out the words other stuff, but have that get redirected to a new file called file2.txt. And you'll see that I ran the command and file2.txt magically appeared in the file manager. And actually, if I want to look at those files, it's as simple as double clicking on one of the files, and it brings up the file in a text editor. The same action could be done at the command line using the cat command. If I say cat file1.txt, it prints out the contents of file1. And so I'm going to close that text editor and just create a couple of more files. So I now have 4 files. And yeah, I could go and look at the files one by one, but that starts to get a little bit tedious if I'm going to do that. So the other way to look at the contents of these files would be to use the cat command, but then use some kind of a wildcard to say print out all the files at once. So I could say something like this. Use the cat command to print out the contents of all files that match this pattern. FILE star is the wildcard, and then .txt. All of these file names match that pattern. And you'll see here, in one step, I've printed out all 4 of those files. Now we're starting to get a bit of the idea of why the command line is so important. If I'm going to do something on one file, it's easier to just click on the file and work on it. If I want to do something on 100 files, though, or 1,000 files, this is where the command line starts to be absolutely essential. And these are the kinds of problems that you will encounter in bioinformatics. You want to work on many files at a time. And so this is why we spend so much time on the command line, because when you're doing serious scientific work, you're going to need it. So the last thing that I want to cover in this section is to talk a little bit about environment variables. So environment variables are convenient ways of storing a short piece of text, some kind of a location of a file, or some kind of a setting for the system. And so let's give some examples. You know that we're using a shell called bash. Bash is the program that interprets the commands that we type in the terminal window. And so there's an environment variable that tells which shell we're using. And that environment variable is called the shell variable, and it's in all capital letters. So to see the value of the shell variable, I type echo $shell. I have to do this all in capital letters, and it returns to me the value of that variable. Now, the $sign, by the way, tells the shell this next thing you're going to get is the name of a variable. Run the echo command and substitute into the command the value of the $shell variable. So in this case, the value of the variable is /bin/bash. That is actually telling the location of the bash program. So bash is a program that resides in the /bin directory, and so the shell is telling where the program is to use to interpret the commands that we type. As I mentioned, the $sign is needed to tell the shell this is an environment variable. If I had typed echo shell without a $sign, it just prints out the word shell. So I need to use that $sign anytime I want to tell the shell this is an environment variable. So there are lots of environment variables in any Unix system. Another one is the path variable. So the path contains a string which lists all of the directories in which the shell is going to search to look for a program. And so whether it be the echo program or the cat program or the ls program, all of those are in a directory called /bin, or I think they're /bin. Some of them are actually in user bin, and that would be, where are we now? We have a whole bunch of these. Okay, there we go. There's it's actually split on a couple of lines. I'm going to just make this bigger so maybe we'll fit all of that. So here are in the path /user bin and /bin. So those are most of the Unix commands you might type. There are some that are installed locally on the system. They don't come with the out-of-the-box system. That's user local bin. So there are more commands there. And then for the Birch system, we have a number of directories that have the bioinformatics programs that we work with. Home psgendb local script has scripts for the Birch system, and home psgendb local bin Linux x86-64 has executable binaries. And so all of these are places where the shell will look to find a program whose name you type at the command line. If the program isn't found in any of these directories, it will give you a command not found message. So there's even an environment variable for the Birch system. That's $birch. And so the location of the Birch system is found in this /home/psgendb PSGENDB directory, so the actual location would be different on different systems depending where Birch is installed. But I can always find out where Birch is installed just by typing echo Birch. So you'll find, oh, maybe one more. If I was going to do BLAST searches, the programs need to know where the BLAST databases are stored. Echo $blast DB tells the location of the BLAST databases. In this case, the BLAST databases are found in a directory called GenBank, which is a subdirectory of /home/PSGENDB. So there are lots of environment variables. They're useful to know about, and as you learn more about Linux, you will actually be using environment variables quite commonly. So finally, I want to say that we haven't covered every Linux command you might ever want to know about, but we've covered quite a lot of them. And this list is what I'll call a short list of Linux commands that are quite useful. And usually to get by at the command line, you only need to know maybe about 15 or 20 commands. And so even though there are hundreds of commands out on the system, you usually only need to learn a few of them. So we've seen the cat command and the cd command, the copy command. As you learn more about Linux, you're going to learn how to use some of these commands. And here's a great link to a Unix tutorial at Duke University, which is one of the best that I've seen. And so I recommend for my students and my courses to do this tutorial. It just takes you step by step through many of the Unix commands. But this is something that would be useful for almost anybody that wants to learn how to work at the Unix command line. So Linux always any Linux system or Unix system will have commands that are shown in these tutorials, and so you would be able to get a good start that way. There are lots of books on using Unix that will tell you more than you ever wanted to learn about Unix commands, and if you just Google Unix tutorial or something like that, you'll find dozens and dozens of tutorials where people are teaching you how to use the command line. There are lots of videos as well on YouTube that will do much the same thing. And so this is, though, a good entry point to learning how to use Unix. So by now you're probably wondering, well, where do I go from here? How do I get started in using Linux and bioinformatics? And so I have a number of things for people, first of all, at the University of Manitoba, and then if you're not at the U of M, I'll give you some more general guidelines. So for people like my own students in my courses, the first thing to do is to go to the Birch website at home.cc.umanitoba.ca /PSGENDB. And this will give you some links to, first of all, connecting using ThinLinc, connecting to the servers, setting up your account, so there's a first time setup process, and then how to customize your screen. And once you've got your Linux account set up, you'll be ready to do the tutorials on the Birch website that take you into the many different things that you might want to do in bioinformatics, whether that's genomics tasks or cloning tasks or anything you might do with sequences or sequence databases, phylogeny, multiple alignment, sequence database searches. There's quite a large number of tutorials on this system that will teach you how to use Birch. And I know that some people viewing this video might not be from the University of Manitoba, and so I've deliberately set things up to give you some introductions. There are more Linux distributions out there than you can count, but my favorite is the Ubuntu Mate Linux, and there's the URL for Ubuntu Mate. And so this is probably one of the easiest distributions to use if you're getting a start in Linux. The easiest way to do that is find a computer that somebody's thrown away or that's too obsolete to use, wipe the disk, and install Linux on it. You can download Linux and have it install on your system. It's very easy to do. Most of these websites will give you instructions for how to do that across the network. You need a good network connection to do this, and I strongly advise doing that, by the way, on a hardwired Ethernet if you at all possibly can, but it is possible to do on Wi-Fi. And once you've done that, you would want to install the Birch system, which is available for Linux or Mac OS X or OS X. And again, that's downloadable from the Birch website, for which I have the URL here. And from that point, then, you can do the first time setup on your system and customizing your desktop, and then learning bioinformatics through the tutorials. Now, I'm telling you right now about how to get started on a desktop machine, but again, if this is just a desktop machine, you'll still have the hardware limitations. You might only have maybe 4 CPUs or maybe 8 if it's a more recent machine. If you just bought the computer, it would probably have maybe 8 or 16 gigabytes of data. The more of RAM, the more RAM, the better. If it's an older machine, fewer of the bioinformatics tasks will work, although you can do almost all the tutorials with just 8 gigabytes of RAM. That's most of the Birch tutorials will work even with that low amount of memory. If you are setting up a system for a department or an institute, then you need to talk about getting some kind of a server that will have maybe at least 64 gigs of RAM, or maybe even better, something like 256 gigabytes of RAM, multiple CPUs, probably at least about I don't know, 32 or 48 CPUs, or even more than that, if you can afford it. The kinds of systems I'm talking about, you can get started on for probably around $25,000. So this is more of a departmental type purchase rather than something you would put in your own lab. But the point is that sounds like a lot from the perspective of somebody who's bought a desktop computer, but if you're creating a server, that's really a pretty minimal amount of money. And for that, you will then have a server that you can that will do work for an entire department and can serve multiple users at a time, to get some pretty sophisticated tasks done in bioinformatics. So I'll just give a final plug for the Birch system. It's downloadable at the Birch website. And when you use this system, please remember that you're getting it for free, but the way you pay for free open source software is by citing it. And so here's the citation for the Birch paper. And I want to thank all of you for listening, and I hope you try the Birch system and get into Linux and find all of the wonderful things that Linux can do for you in bioinformatics.